All right, today we're jumping into this incredible story of, well, biological espionage. We're going to uncover how scientists figured out how to tame a tiny little plant pathogen and turn it into one of the most powerful tools in all of modern biotechnology. So it all starts with what sounds like a pretty simple question, right? If you have a gene for, say, drought resistance or better nutrition, how do you actually get it inside a plant cell and make it a permanent part of that plant's DNA? I mean, that's a huge technical challenge. But as it turns out, we didn't have to invent a solution completely from scratch. And you're not going to believe this, but the key to modern genetic engineering actually came from studying this bizarre plant disease. These growths, they're called crown galls, and they're basically plant tumors. But they're not caused by a virus or a fungus. Nope. They're caused by a bacterium with a very, very special set of skills. So let's meet our main character, Agrobacterium tumefaciens. It's a super common soil bacterium that scientists have nicknamed nature's genetic engineer. And for good reason. It has this incredible ability to literally hack into the plant kingdom. So how does it do it? Well, when a plant gets a wound, Agrobacterium senses it and moves in. But it's not just a simple infection. Oh no. It triggers this rapid, uncontrolled cell division, and that's what creates that tumor-like crown gall. What's happening on a molecular level, though? That's even crazier.
Yeah, this isn't just some disease. The bacterium is pulling off a seriously sophisticated act of genetic manipulation. It's actively rewriting the plant's own DNA for its own benefit. It's a true genetic heist.
So you're probably wondering, how does this tiny bacterium pull off such a complex operation? Well, it has a secret weapon, a specialized piece of DNA that's basically its toolkit for invasion. And here it is. The bacterium's secret weapon is this large, circular piece of DNA called the T plasmid. That stands for tumor-inducing. You can think of this whole plasmid like a USB drive. And that red section, the tDNA, that's the malicious software it's designed to deliver. Now here's the key step, the real magic. The bacterium finds the plant's DNA and physically injects its tDNA. And that tDNA integrates itself right into the plant's chromosome. From that moment on, the plant treats this foreign bacterial DNA as its own. It just reads the instructions and carries them out. The heist is complete. So what's actually on this malicious software? Well, the tDNA has two main sets of instructions. First, it has oncogenes, basically cancer genes, that tell the plant cells to just divide like crazy, and that's what creates the tumor. Second, it has genes that force the plant to produce these really unique compounds called opines. And opines are a specialized food source that only the bacterium can eat. It literally turns the plant into its own personal lunch factory. So when scientists saw this incredibly efficient gene transfer system in action, they had a brilliant idea. What if? What if they could hijack the hijacker? And this, right here, was the turning point that transformed this plant pathogen from a foe into a friend. This was the big aha moment. What if we could keep the delivery system, the bacterium's amazing ability to inject DNA, but just swap out the cargo? Get rid of the tumor genes, get rid of the opine genes, and put something beneficial in their place instead. The challenge was pretty clear. The original Ti plasmid is basically a dangerous weapon. It's also massive. We're talking over 200,000 base pairs long, which makes it incredibly difficult to work with in a lab. So the goal was to engineer a new version. Something smaller, safer, and way more useful. A tool we now call a vector. And the process for doing this is called disarming. Scientists basically used molecular scissors to just snip out the genes that cause tumor growth, the ones that produce plant hormones like auxin and cytokinin. And they also took out the genes for making that special bacteria food. The key was to remove all the harmful code without breaking the machinery that actually transfers the DNA. And this is the result, a disarmed plasmid. The nasty tumor-causing genes are gone. All that's left of the original tDNA are these little bookends, the left and right border sequences. And these borders act like punctuation marks, telling the bacterium, okay, cut here and insert everything in between. That space is now a totally empty cargo hold, just waiting for us to load it with any gene we want. Okay, so once the basic concept was proven, the next step was to refine the tool, right? Make it even easier to use. And this led to two main strategies for building these new genetic vectors.
The early efforts used what's called a co-integrative system. It involved merging a gene into one massive, disarmed T plasmid inside the bacterium. It worked, sure, but it was really clunky. The real breakthrough, the real game changer, was the binary system, which took more of a divide and conquer approach. And here's the beauty of the binary system. It's so elegant. You have two separate plasmids living in the same agrobacterium. Plasmid A is the big helper plasmid. It has all the Vir genes, that's the machinery needed to actually perform the DNA transfer, but it has no tDNA to send. Then there's plasmid B, a small, easy-to-handle vector that contains our desired gene all neatly packaged between those tDNA border sequences. The helper plasmid's machinery sees the border sequences on the small plasmid and sends that into the plant. It's a perfect division of labor. And the advantages really pop when you compare them side by side. Co-integrative vectors are huge and require this tricky recombination step inside the bacteria. The binary system, with its two smaller plasmids, no recombination needed, and it's just way, way easier to work with. So it's really no surprise that it became the industry standard. So this binary vector system really is the foundation of the modern toolkit for plant genetic engineering. Let's just quickly look at why it's so powerful and effective. You know, the advantages of the binary system are just so clear. The smaller plasmids are much easier to modify and to multiply, which just makes for a much smoother workflow in the lab. This system also lets scientists do all the heavy lifting, all the cloning and preparation in a common, fast-growing lab workhorse, E. coli. This ability makes the small plasmid what we call a shuttle vector. It's designed with two different origins of replication so it can survive and be copied in both E. coli and agrobacterium. This means you can prepare millions of copies of your gene in E. coli, and then you simply shuttle the finished plasmid over to agrobacterium for the final delivery into the plant. It's just so efficient. So the whole modern process really just boils down to these three simple steps. First, you clone your gene of interest into that small shuttle vector, usually in E. coli. Second, you move that finished vector into the agrobacterium that's already got the helper plasmid waiting. And third, you just let the tamed bacterium do what it does best, find a plant cell and deliver its genetic payload. And that really brings us to today. By understanding and disarming a natural plant disease, we have developed this incredibly precise tool to edit the very blueprint of life. We've gone from being the victims of a bacterial hacker to using its own code to improve our world. The only question left is, what do we want to write into that code next?